Hello.
Hi.
How are you?
Anybody else around?
Yeah, well, I heard from maybe one or two people. They might show up. There's, just people are trying to get back into the new year, I guess.
Yeah. I've got a paper here that I've been reading that is giving me a headache, so yeah. Maybe I'll, if you want it in the chat, it's about tensegrity, of course.
Oh, yeah, that'd be great.
It's Journal of Mechanics and Physics of Solids.
Okay.
And then it's on the mechanical modeling of the extreme softening/stiffening response of auxiliary loaded tensegrity prisms. Anyway, I'll find it and put it in the chat here.
There we go.
All right. And then you show the PowerPoint.
There we go.
So I start off with this. Actually, I kind of wanted to show you this. I turned this upside down, but this is the cytoskeleton is represented here by microfilaments. And then you have this, and this is a soft surface, and you end up with some microtubules and a bunch of microfilaments across here.
Yeah.
And desmosomes, which don't seem to attach to too much. And then there's a bit going on at the bottom. So I guess this is soft. There's really just some attachments at the bottom.
Okay.
And then this is a stiff surface, and a lot happening with the substrate, extracellular matrix at the bottom.
Okay.
So this is kind of what I was going with an attachment at the top and attachment at the bottom, and varying the bottom, the ECM. Anyway, so that's sort of what I was modeling, and then I found this paper, and there's the reference here.
Okay, there
I'll
it is.
put this in the chat. Okay, type in chat. Posted, okay. And then back to this. They just tried it with this simple 3-strut tensegrity. So,
okay.
Yeah. And it moves between the strings touching and the bars touching, basically. So this is the top, looking down at the top of it, and this is the side. So it does change height, and it twists when it moves down. And you can see from this that they have a twist on the triangle. Like the triangles are not aligned; they're twisted. So this is a twisted version. A straight version doesn't tend to stand up by itself.
Hey, Susan?
Yeah?
Yeah, a small mathematical question. Has anyone proven that is the minimal tensegrity structure?
That this is a what?
The minimal tensegrity structure.
Oh, okay. You can do 2D ones.
You can do a 2D one. Okay,
yeah.
so
it's
not the minimal.
So this is a minimal prism model. Okay?
Okay, what's the definition of a prism?
3D structure.
Okay. If you're not living in flatland.
Yeah.
Yeah, I don't know other than that.
Okay, sorry to quibble, but I have a degree in mathematics.
Yeah, I don't have a degree in mathematics. I like math, but that's about as far as it goes.
Okay.
And this is an interesting curve here. If you have a short model, like instead of a long, tall one, short, or they're calling it the thick model, and you have elastic strings, you get these curves for different rigidity.
Yeah.
And if you have rigid cables, you get your J curve.
Okay.
And it's dependent on the base. Alpha and beta are the rigidity multipliers of the strings and the bars. So
which curve is a J curve?
well, these are J curves, except
that I see.
they're not
Yeah.
J curves.
Those are called J curves. Okay.
And
Okay.
if you take the derivative of these in certain ways, you also get a J curve. So I suspect they're kind of an exponential curve. Yeah. Anyway, this is what they were getting with their modeling. And there's your J curve. They're using rigid cables, just like Stephen Levin said. So I'm going, okay. This is in compression, though, and this is in tension.
Okay.
And they all seem to follow the same curve in tension, which is really interesting, except for the ones that are basically loose, that are not really attached. Alpha equals 1, beta equals 1. They're very soft materials. It's this darker blue. I don't know, I thought these were really great curves, but they didn't do what I wanted them to do, so I hope I can get a hold of their data so that I can do my own thing with their data.
Oh.
I don't know if they even have it available. So
soft curves could be what we call going to mush.
yes, or an embryo starting out or something.
Okay.
At all times, this alpha equals 1, beta equals 1 is a completely different curve from the other ones. It's interesting. And then this is the tall model, and they actually made them. This is one way to make a tensegrity structure, kind of nice. They kind of give you, and by showing this, they show you how you could do it yourself. But the curves are like this, with the slender model with the rigid cables in compression. And then you have your J curves again, except these are different from the higher tensions. And of course, the blue curve, alpha equals 1, beta equals 1, is totally different. It goes its own way, we'll just say. And then this one is the dotted lines are where the pre-strain is. They're looking at various pre-strains. That's the P0, and the rigid and elastic cross strings. So the rigid cross strings give you a sort of a J curve here, and the elastic strings give you this kind of thing happening. Sort of a J curve sometimes, sort of not. So this would make a difference if it was in a cell.
Yeah. Susan, can I put on your engineering hat?
Okay.
Okay. Are you familiar with space needles?
I've heard of them.
Okay. The idea is to build a structure that goes from the ground into outer space, so you just have to go up and down it like an elevator.
Oh, okay.
Instead of launching rockets.
Yeah.
Okay. Can you make a tensegrity space needle?
Maybe I'll just say it's more possible with the tensegrity structure.
Unless it flops over like the dinosaur necks that Steve Levin showed.
Oh, well, they might be floppy under certain tensions. There's a collapse.
That's what I'm saying. The idea was to make them out of very long carbon nanotubes, but people proposing them didn't seem to know anything about tensegrity.
Oh, well, that could be a problem.
Yeah, well, that's why I'm suggesting. Maybe there's a solution in tensegrity.
Well, there might be.
There might be, yeah. I don't know if you can make it.
I'm having trouble reading the last part of this lovely paper.
You need a 100-kilometer long tensegrity structure.
Yeah, if it flopped, it would be quite something.
Yes. Maybe with the rigid components being nanotubes. I don't know.
I don't know.
Okay, Bradly's ready to write the paper.
Yeah.
There is a point of collapse that this paper points out. So I'll quit sharing here. That's kind of all I got to.
Okay.
Then I ran into the paper doing further research, this being further research with a lovely derivative. These are derivative curves. So
Okay.
I'm just going to say, okay, fine,
Okay.
like that.
Okay, can you send me that paper, please?
Sure.
Okay, thanks.
Stop sharing. That would be good. Okay, there.
So
Oh,
what are the differences between stiffness and elastic components of the network? So I think they modeled both in that paper. We talked about that
Yeah.
with respect to biology, how you actually have a lot of elastic components as opposed to stiff components. What's the difference?
And apparently in a tensegrity, you need stiff components.
Yeah.
And
okay.
if you look at some of the papers about actin, they have they're rather stiff. Like their intention, they're quite stiff.
Susan, can you make a tensegrity structure with all stiff components?
Yes.
You can. Okay.
I did as Stephen Levin suggested with this, and I put
Oh,
fishing line on it, and if you compress it like this, you can see.
oops, you're not on camera.
Yeah, I'm compressing it on the side, and it's moving in all directions. It's compressing in all directions.
Yeah.
And if you pull on it, the same thing
Okay.
happens. If you pull on it, and it expands in all directions. I don't know if you can see.
Okay, what if you replace the fish line by more dowels?
Then it's just a rigid structure. It becomes not a tensegrity exactly, although there's the X model where you have a rigid X
Yeah.
construct in the middle of
Sure
it.
of that.
Pardon?
Are
Am I
you
sure of that?
It seems to me that the paper you just showed, at some extremes, the elastic components are replaced by rods.
No, they replaced them by a rigid cable. They're cables. They don't
What's
have.
a rigid? Yeah, but what's a rigid cable? Oh, it can't support compression.
Yeah, that's all. You just can't.
Oh, I see.
So I have some material here. I was going to make my own tensegrity as you see.
Yeah, but I'm saying it might be interesting to replace them by rods.
Well, then you just got a geodesic dome or something.
Yeah, I suppose, but it's an extreme example
Well, yeah.
because even a rod has some compressibility and some stretchability.
Yeah, they do. Well, I was leaving
They
that
do. So
out alone.
yeah,
in my
so you
mind.
get the extremes of their parameters, but still, it might be worth looking at. I don't know.
Yeah, well, I know in diabetes they have, for instance, softer cells. So I was wondering if you could relate a diabetic model to the elastic models. So they have a slightly different behavior than the. Anyway, whatever. I mean, it's just an application. I'm not Anyway, I do need to understand this paper, and I need to use the model I'm doing to get some of these parameters. And
in terms of the spacing you look at, do you know if there's any way of making a random mixture of carbon nanotubes so that the connections between the carbon nanotubes are as strong as the nanotubes themselves?
okay, I just saw some papers on that, but of course I didn't download them.
Of course.
Yeah, there's the interconnections you can make with them, but
Oh, okay.
that's all I know.
Okay.
Yeah.
So someday, yes, I can see it now. You know where you can sell them? As bats for baseball.
Oh, okay.
A tensegrity bat.
Oh, I wonder how well that would go over. Tensegrity. Yeah, like an advertising campaign. Yeah.
It flies, the ball flies.
Except when the bat collapses.
Well, you know, that reminds me of the U.S. used to send guns up to northern Canada for testing, and they made them out of plastic, and the Canadians would just take them and crack them.
Yeah.
For the cold, yeah.
That's why the Ford sends a whole bunch of trial vehicles up to the paw. Be 70 below, the bumpers fall off or something.
Okay.
Yeah, plastic doesn't do cold.
Yeah.
I know that. I've dropped things out of our freezer.
Okay, well, I accidentally bumped into somebody behind me, and she had this beautiful plexiglass square thing right in front of her truck. And I'm thinking, why?
A shattering experience.
Yes.
There we go. You have to do the thermal properties of tensegrity structures.
Yeah.
Right. In that case, I'll make them out of string.
Yeah.
Yes.
Oh, I do have some rods and some ends and stuff over here that I'm going to make.
Yeah, you just take them outside in February. we typically go to minus 40, occasionally minus 50. Yeah.
Okay. Yeah,
Okay,
wow.
but this is steel cable. I think it'll work.
You think it'll work even at minus 40, huh?
Yes. If it isn't minus 80, we'll be good.
Okay. Well, we'll send you up north for experiments.
Yeah. I'll go visit my nephew. He lives there.
Okay.
Yeah. So yeah, what were you going to do today, Bradley?
Oh, well,
I had on the thermal properties of tensegrity structure.
yeah. I had some interest in the meeting from a couple people, but I don't know if they're going to make it. So in the Slack, they'd mentioned it, but I don't think. In any case, I'm going to go over a couple things today. I have there's some new project, we're going to have a project for this summer of code, and I'm going to go over a little bit about that. And then I'm going to go over the Learning on Graphs conference, which I wrote a blog post on it. And that was one of the conferences I talked about in the last meeting. And I kind of prepared a whole review of it. And then I'll probably go over a paper. So let me share my screen. All right. There, you can see my screen. Okay. So the first thing I want to talk about is this project. So right around January, they always request the Summer of Code projects. We're sponsored by INCF, which is the Neuroinformatics Core. They sponsor our projects, and so this is the project for this year I've come up with. We're only going to, I think, do one this year. And I talked to Jia Hong, who did the Graph Neural Networks project last year, and he's interested in mentoring this project with me, being a co-mentor. And that's good because he did a lot of the work on building this pipeline that is mentioned in this project. So this is the Graph Neural Networks project that we did last year, but it's a different version of it. So last year we did the Graph Neural Networks project. We wanted to get into it and see how far we could go with this tool called Graph Neural Networks. Basically, you're taking input data maybe from microscopy and you're forming a graph out of it. So any kind of data that has graphical structure to it, you can use for a graph neural network. And that's fortunate because there's a lot of data in the world that forms a graph in one way or another. It could be just, points in a space that can be connected either geometrically or through some statistical association, or it could be, like a tensegrity network, or it could be other things. So what we're interested in here is building a neural network that can process the structure of the, in this case, it would be microscopy images, at least for this pipeline, and then it would produce these graph embeddings, which are representations of that structure in the image that would then be produced as a computational structure on the other end. So it's basically processing things in the image. It's grabbing landmarks, it's putting those together, and then it's finding the common graph representations or embeddings that exist in that image, in that data set. So, you have a lot of variation in the input data. You want to find the common embeddings that exist for it. So that's what graph neural networks do. And so in this project, so last year we did this pipeline where we sort of took DevoLearn, which is our existing software that we have, and we refactored part of it or rewrote it to sort of optimize it for this sort of application. So there were a number of updates that needed to be done, and that was Wataru who did a lot of that work. And so that part of the pipeline was completed. We also had another part of the pipeline was completed, which was to implement a neural network that could do this type of transformation. So that was Jia Hong, and there was another person, I can't remember his name right now, but he was involved in the project. And Jia Hong was the person who kind of laid out this pipeline of processing the data to building this neural network and benchmarking it, and then to this other third step, which wasn't completed, which was topological data analysis and higher order data analysis. So this is the thing we're going to focus on this summer. So, these graph neural networks are actually really good at figuring out shape transformations, which are actually in embryogenesis is very important because you have a lot of things changing shape. You have a lot of things, cells moving around and things like that. So, this is one thing we want to capture. So we have this ability to segment raw data and incorporate it into this pipeline, refining the method with respect to deriving graph embeddings, and then, refactoring the models, and then extending this to topological data analysis and complex network theory. So this is these are two output areas. So once we get these neural networks that can do these transformations and produce these embeddings, then we can go on and do like topological data analysis. So we can look at like the system in different states. You'll have all these embeddings that get produced that we can then analyze. we can compare the structures using some sort of unified method. And so this project involves doing a lot of this maintenance work, like refactoring where needed, basically taking the model, running through it, refactoring things where needed, maintaining it, and then focusing on this third aspect, which is to sort of figure out how to use the output. And so that's what this project is about. And it's kind of hard to get your head around it. Like I found that the students, it was kind of hard for them to get their heads around it at first, because there is a lot here that's, there's a steep learning curve for this sort of graph neural network work.
Okay, Bradley?
Yeah.
I put a name in the chat.
Okay.
Would you please include her on the mailing list for possible summer students?
Okay.
Okay, she wrote to me yesterday, and I didn't think of anything, but of course you're a responsibility.
Okay. Yeah.
Okay.
All right. Did they contact you, or?
Yeah, she contacted me. She's the daughter of a lady I work with on bubble farming.
Okay. Yeah. Okay. Yeah.
She's into all sorts of things which would overlap with you a lot.
Okay, it sounds good. Yeah.
Okay.
Yeah. I'll get in contact with that person.
Okay.
Yeah.
She might be interested in doing that.
Yeah. So that's our project for Summer of Code. And, I think we're supposed to submit them by the end of the month. So this will go through maybe a couple more revisions, and then we'll put up the project and we'll have a call for involvement. And yeah, people can, if they're interested in participating, they can go to some of these. So some of these things are drawing from, of course, we have the website with, we have some papers. We don't really have much on graph neural networks right now, but you can get a sense of what we're doing in the group. We have the DevoGraph software on GitHub. So we have our DevoLearn repository, which involves a lot of the work we've done with deep learning. And in the DevoLearn repository, we have DevoGraph, which is basically, last fall I took a lot of the stuff from the Google Summer Code project and put it into the, published it into the repo. So this is the DevoGraph repo here. You can go in and download the software and run it. So this is open source under MIT license. You can run the software and see where we are with it. And of course, I would point potential students to this because it allows them to sort of identify maybe what they could do if they can't, if there are things that they see that are problematic when they try to run the software, or they think that there are other things that need to be done. There needs to be some, I think, more documentation as well. So that's another thing that can be done in the project period, is to do some documentation. They don't often talk about that, but that's very important to get people to understand what's going on, because it's a very hard thing to really get right off the bat. So that's going to be part of that. And then going through some of our other things that we've done. And then I actually published this blog post, which we'll talk about in a minute. This is us Learning on Graphs conference recap. So yeah, the project uses PyTorch, and if you have experience with Python, then you're probably you're ready to go with this. Any work with PyTorch or TensorFlow is good. If you know how to use a workflow there, that's good, because this is going to require a lot of the tools that they have there. I think they have toolboxes for graph neural networks, so that will help. And then being able to work with data sets. So last summer we had a couple of benchmark data sets that we used. I don't know if they're in this repository. They're in this data directory. And I think the data sets are in here. So the EPIC data set was used, which is a C. elegans data set. this Fluo N2DH Sim Plus. That was another data set. IPSM and this other one here. I'm not sure. I don't know. Well, anyways, we can go, and then there's some raw data. So these can be benchmarked. We may have to go back and kind of annotate what these are, seeing already kind of things that we need to improve upon to get into the to get this repository in shape. So that's all part of the fun, though. It's all part of the fun. Yeah. So more to come on that. So the next thing I want to talk about is this Learning on Graphs conference recap. So last meeting I talked about Learning on Graphs, this conference that happened in early December. And is a nice conference. This is the first conference that I'm aware of that had to do with graph neural networks. I think there have been a lot of workshops at the machine learning conferences, but not a standalone conference. So this was a nice tour of sort of the diversity of research in graph neural networks. So, graph neural networks are, as I described before, you have a lot of graphical input in the world to take things that can form graphs and to transform them into graph embeddings, which you can use for computational analysis. And so one of the interesting talks, one of the first talks was on applying this. In fact, they had a lot of talks where they applied graph neural networks to medicine. But in this case, they applied it to molecular problems and specifically protein folding and protein, working with proteins, basically, different types of prediction tasks. So actually, as it turns out, graph neural networks have a lot of applications in the medical domain. So you can look at things like a molecule where you have this sort of graphical structure to it, where you have other types of biological things with graph structure. I think some of the images that Susan showed of the graph of the cell with a lot of the actin molecules and things like that interconnect spatially, those are very good graph neural network problems, I would assume, because you have a lot of those components that are sort of form a graph. You have to abstract away a lot of the biology, but it becomes useful as a graph problem. So, there were some really a lot of interesting talks in medicine, not so much on biology, like on bioengineering. But yeah, there are a lot of, I think people are really pushing this in terms of drug discovery and characterizing molecular systems.
Okay. Bradley, when you have a break, I've got a question that's sort of related.
Oh, go ahead.
Oh, okay. This is a little crazy, but I wrote a paper many decades ago on linking Christmas tree light bulbs in series.
Okay.
Okay. And I tried to solve the mathematics for them. I actually did experiments with them. I couldn't get beyond 2 light bulbs from the mathematics. It was too complicated.
Yeah.
Basically, if you have a string of light bulbs in series and all of them are blinking, if one goes off, they all go off. So the thermal component that turns them on and off has to cool for all of them simultaneously. And then the first one, then when they're all on, you get a circuit again. Okay? And they all light up. Okay?
Yeah.
Empirically, I found that they slowed down as you added more and more light bulbs.
Okay.
Okay. Now here's my question, which came up this Christmas. Okay. Well, But if you take a network and at each node you place a blinking Christmas tree light bulb, and then you take 2 nodes and you pass current from one to the other, what happens?
Right.
Okay. Do you get a short circuit, or do different parts of the network blink on or off?
Yeah.
It depends on whether it's an LED and it's not, therefore it's a diode and only passes current in one direction.
I did these experiments before LEDs were invented.
Okay.
Okay.
They each light bulb had a thermal switch, so the current would go through, the thermal switch would open, and the current would stop because it heated up.
Yeah.
Okay, I have no idea how LEDs work.
They're a diode, so
These were simple thermal switches like you find in aquarium heaters.
oh, okay. Anyway, LED just will just allow current to flow in one direction.
What turns it off?
A lack of current. So,
that's not good enough.
so you can put a multi-vibrator circuit in, like a 555.
Oh, yeah. Well, then there is a kind of vibrator. That's cheating.
Okay, it's cheating.
Because each one does not affect all the others.
Right.
Except that if it doesn't get any current, I suppose. Well, okay, maybe it's a different case.
Yeah.
At any rate.
Actually, yeah.
You have any? Okay, Susan, you studied electrical engineering.
Yes.
If you pass the current through an arbitrary graph from one node to another one on the other side, are you going to get a short circuit?
Yeah.
Depends on your definition of a short circuit.
The whole thing blows up.
If the lights have a resistance, then no.
No.
But if the lights
some
don't,
paths through the network will be longer than others.
yeah, so you're going to have some resistance there. It's just how much resistance do you
Okay. Okay.
stop the wires from melting.
The deadline is next December 25th.
Okay.
Yeah.
So, yeah, I mean,
Have you ever seen anything like that, Bradley? Passing current through the network?
well, not really. I mean, I've heard of people have analyzed power grids and things like that where there's a flow of electricity and then there's a disruption, and you can look at like cascading failures and things like that.
Right.
Then there's like synchronization work on like fireflies and things like that, but the network there is different.
Yeah.
So, right.
That's different because each one's perceiving the other and trying to train with it. Okay.
Yeah.
Okay. Yeah. This is each one can, if it's just a single line, each one can literally turn them all off.
Oh yeah. Yeah.
Okay. But if it's a whole network, what happens?
Right. Right.
Okay.
Yeah.
So I think it'll be an amusing paper to consider.
Yeah. So that actually brings up an interesting point with GNNs, because they actually do this thing with what they call message passing. And message passing is a technique that they use to look at, like, to build the network. So they have a series of inferential techniques to build these networks. Because as I said, in the real world, you have these features in an image or something like that. And there's a graph structure, but you have to find the graph structure. You have to discover it. Because you have to train the network. So the one way they do this is through message passing, which is something that is from like communication networks, but actually works with electrical currents. So you're passing things from node to node, which could be a message, it could be electricity, and then that allows you to approximate the network. And so that, in that sense,
look, I got to go to another important meeting. It's called coffee.
Okay. Yeah. Yeah.
Okay. Have a good day.
You too.
Bye.
See you later. Bye. So this is one of the inferential techniques that they use. It's very common in the field, and there are other. I mean, the point of this blog post is to point out some of the contemporary things going on in this field, but there's a lot of opportunity to innovate and to find like new approaches to these things, because message passing is good, but it has its limits. And of course, if you're dealing with different types of data, like biological data of different types, you may need to use other techniques. So some of these other techniques are or maybe inspired by differential geometry and algebraic topology. These would be continuous methods, whereas message passing is more of a usually thought of as a discrete technique.
It's a digital technique then,
Yeah.
the message passing?
Yeah, because
Okay.
you're passive.
There is an underlying sort of pulse in the brain. I'm sure it's due to the way the arteries and veins work in the brain, and it gives it's a sort of an interesting fractal problem, similar to some of these networks, I guess, because you'll get I don't know if you have valves there or not, but it takes a while for the brain to be perfused with blood. So anyway, there's some sort of an underlying pulse that goes on.
Okay, yeah.
Actually physical. And there's also an electrical pulse. So I was just kind of intrigued by all of that.
Yeah.
There's somebody studying the underlying electrical pulse in the brain at the University of Manitoba, and I asked him about ventilator injury and the fact that a ventilator has a mechanical pump and it just pumps regularly, but whereas your normal breathing is kind of fractal and it gets into the lungs and therefore gets oxygen to all parts of the lungs and your body better if the pump is run in a more fractal manner. So it all fits together here.
Yeah.
You know, in networks, some of this would fit together.
Yeah. Yeah.
I don't know if you think it applies, but it's the same type of thing.
Yeah. Yeah.
I'm interested in the graphs.
Yeah.
Partly because it will solve the tensegrity structure for me too. Because tensegrity structures are a graph, basically.
Right. So yeah, one of the things that one of the things I noticed about a lot of these techniques, and there are a lot of techniques here that, so there's this gap, I think, in the literature as to how to link these to real world problems. So like if you're taking data from some source, the graph structure, they can be very diverse. And they say, well, these are generalized models and you can just, analyze the graph with like all these tools. And there are all sorts of, isomorphism tests to say like if two graphs are isomorphic, or, you have other types of tests to look at the sort of to sort of infer the structure of these graphs. But the idea being is that there's a graph in the real world that you can analyze and that they're all universal. And maybe some of these graphs aren't universal. Maybe you need to have like specialized graph neural networks for different types of networks, whether they be like an electrical pulse, a set of electrical pulses in a network, or a set of forces in a network, or something more abstract like statistical associations. Or I think there's something called graphons, which they talk about here, which are alternate graph representations that are sort of, where you get these different graphs that form, but they're all basically related to one another, and you can treat them similarly mathematically. So there's a lot of diversity there in terms of what we're dealing with graphs. And so I think this approach is widely applicable, but you also have to think about the system that you're dealing with. And there are a lot of systems that form graphs, very widely construed. So like tensegrity networks, that would be.
Yeah.
So, so then I guess to sum up here, there's this Cats for AI workshop, which is a workshop that is hosted in October. This is maybe the precursor to this conference. This kind of goes over category theory and artificial intelligence, and you say, what does category theory have to do with networks? And the answer is that you can use category theory in really interesting ways to sort of define the networks that you're going to be analyzing. And this has links to artificial intelligence and some of the classification tasks that you're doing there. So there's this whole sub research field on category theory for AI. And one of the speakers talked about this in depth, Taco Cohen, who is from, I can't remember, from Qualcomm AI. And he talked about some of these tools from category theory and applied math that are, that you can use to analyze the input data in interesting ways and then have a nice output that's amenable to things like causal models. So you can look at causality in the networks, what causes something to happen. And with networks, it's hard to really get a handle on causality because you have a lot of things interacting at once. And so to find these causal arcs, it's really kind of powerful. And then the end of this workshop was just, or the end of the conference was just these workshops. They had each day, at the end of the day, they had workshops. So they had one on neural algorithmic reasoning, which was saying that optimization problems are, hard. It's hard to solve optimization problems, but they can be made easier using these kind of graph neural networks or neural representation learning. Then there was this issue of expressiveness, which was basically how can you cast the net of your method to a series of problems. So if I have a neural network and I'm running it, can I run it on any problem in the world, or can I only run it on a small range of problems? And when you talk about an algorithm's expressiveness, you're talking about how widely you can cast that net. So maybe, there's some network approaches that are only good for looking at, brain networks, things extracted from neuroimaging data, or certain things that are maybe good for power networks, like utilities. But those models are maybe have limited usefulness beyond that. So there's a whole research area on expressive algorithms. And they talked about that. They talked about graph rewiring, which is where they talked about the message passing approach and how you can rewire graphs. This is very important for graphs that grow and change shape. So the things that we might be doing this summer, there's this whole area of graph rewiring that's interesting, that's probably pretty relevant. And then finally, they featured a very practical workshop on implementing these networks in TensorFlow. So there's a TensorFlow framework for graph neural networks. If you want to implement some of these networks, you can use this framework, and it works in TensorFlow. So if you know TensorFlow, you can walk right through it. And so there are some nice tools that are available for this. There's some, interesting concepts. And I think that's something that if people are interested in, we can talk about. Well, we'll have to talk about it for the Summer of Code project, but just in terms of research, talk about it further.
Udemy has a course on or several courses on TensorFlow. If someone needs to educate themselves, like I do, on them, because I don't know them.
Yeah.
But they'll sell you a course for $20 if you can catch one on sale. Otherwise, they're about 100.
Yeah.
Canadian. So.
Yeah,
you can get a course on it.
Yeah.
TensorFlow and neural networks too, for that matter.
Yeah. So the last thing I wanted to talk about today was, we have a lot of papers that are competing for attention here, but one I wanted to talk about was this paper by Jeremy England. I thought we brought up his name in past meetings, but he does some really interesting work with physics and thermodynamics and biology. And there's this, I think he's from England, I can't remember what university, but he published this paper recently on self-organized computation in the far from equilibrium cell. And this is in Biophysics Reviews, I believe. Biophysics Reviews. And this is an interesting paper. So in the the abstract is very short, but actually he's at Georgia Tech. I'm sorry, he's at Georgia Tech now, and he has an affiliation in Israel as well. So there's this the abstract is recent progress in our understanding of the physics of self-organization in active matter has pointed to the possibility of spontaneous collective behaviors that effectively compute things about the patterns in the surrounding pattern environment. Let me zoom in here. Here we describe this process and speculate about its implications for our understanding of the internal organization of a living cell. So this is a pretty broad, general abstract, but basically he's interested in the self-organization of active matter. It's generating these spontaneous collective behaviors. They compute things about patterns in the environment. And then there's this relationship to the internal organization of cells. And so one of the things he does here is he talks about, and this has been studied, this goes back to Schrodinger's paper, What is Life, back in 1944. And this talks about how the cell's whole array of chemical reactions and transduction of molecular signals and the generation of controlled forces somehow happen in obedience to known physical laws, but still it's impossible to imagine how this leads to a living system, or at least according to Schrodinger, because we kind of know some of that now. We have some good ideas anyways. So this is, kind of, we've worked through that since then. And so there's been a lot of biophysics work on this. And, some of these, we know a lot of the properties of some of these molecular mechanisms, for example. We know a lot of things, more things about inheritance and DNA and other types of things that are important to this question. And then this is going through the literature. It turns out that several streams converging from different literatures have begun to point together to add a new answer to this question. So in cell biology, advances in microscopy have revealed an economy of adaptive and multifunctional membraneless structures whose feats of self-assembly may harness a variety of effects of non-equilibrium statistical mechanics. So you're in here, I think you may be talking about some of the things within, within the cell, or things that, self-organized structures that self-organize as adaptive matter. And so finally, progress in our understanding of the physics of active matter suggests that a variety of spontaneous learning and computing behaviors would be surprisingly easy to get for free, which is that's a common theme in complexity theory, this idea of order for free. So self-organization is often referred to as order for free, where the idea is that there's entropy, it contributes to everything decaying, but we have somehow over time we get self-organization of structures, and that at least intuitively shouldn't happen when you have entropy everything subject to entropy. It shouldn't happen, but it does. And so we have to explain this through self-organization. So maybe surprisingly easy to get to for free from the general principles governing self-organization in active mixtures exposed to patterns. So he talks about exposing something to patterns. I'm not sure exactly what that means. Taken together, these distinct sources of evidence and concepts provide a new perspective on biological adaptation. So he's talking about biomolecular self-organization here. So let's see if he doesn't give a number of. So he gives this this measure that he comes up with called the rattling number, and specifically this idea of low rattling. So he draws from things like dissipative structures and other types of physical tools that have been, formulated in the past to sort of explain this. But he's kind of coming up with a new innovation here, which is this rattling number. So this part of the paper, I'm going to kind of go over it a little bit and may give us some insight. Active matter is a class of non-equilibrium many-body systems in which individual particles or components have access to energy from external driving forces. So that could mean like, external energy like the sun or some chemical reactions or something else. A variety of active matter systems ranging from protein motor and filament mixtures, which we've talked about as being part of that macromolecular milieu, to chemically catalytic colloid suspensions. So these are like different particulates that are in suspension. We've talked about things like the self-assembly of different particles, or different types of macromolecules even, I guess, or robot swarms have been studied experimentally. And they are known to exhibit a range of striking collective behaviors, which then the examples are spiral vortices, regular crystals, and synchronized dances have all been observed in instances where many interacting active elements are subjected to non-equilibrium driving. So you have this non-equilibrium energy out in the environment. It's fluctuating in a non-equilibrium state. And then you have the system that's in equilibrium, where at least nominally, where these elements are interacting, and they're being driven by this non-equilibrium driving. So this leads to this sort of fluctuation, this introduced fluctuation into this system of interactions. So this is how, you kind of explain the self-organization. And so active matter may provide an exciting empirical experimental playground for discovering and characterizing interesting non-equilibrium collective phenomena. But of course, it's highly challenging to make a prediction here. So, there are things called fluctuation theorems, which have really kind of driven the progress in this field, where, you have these general results in statistical thermodynamics governing the arbitrary, far from equilibrium and nonlinear regime. So you can use these tools to kind of look at some of these systems. Let's see. Perhaps the most fundamental statement was that of Crookes, who showed that the relative probability of a dynamical trajectory, which is this formula here, or this statement here, and this time-reversed movie. So the dynamical trajectory has a time-reversed movie, which is basically the inverse of that, are related simply to the heat Q, or at least doing the forward trajectory by this equation here. The Crookes relation does not imply a generalization of the minimum entropy production principle that holds in linear non-equilibrium regimes. Indeed, it makes clear instead that no such simple thermodynamic principle must govern all non-equilibrium steady states. So this is a rigorous argument for an exact quantitative relationship between the stability of ordered non-equilibrium structures and the likely amounts of work absorbed during the history of their formation. So you can actually get a sense of the thermodynamic work being done in these ordered non-equilibrium structures. So he talks about, let's see where he talks about the rattling number in here. So the experimental example described, it turns out to be a simple and intuitive instance of a more general phenomenon known as low rattling. So he's talking here in the previous paragraph about this experiment that separates local and global dynamics. So let's see. Okay. Specifically, it can be the case that the same external driving forces acting on a system may activate motion that is more or less orderly, or more or less violent, depending on what state the system starts in while being driven. A simple example is that of living crystals, in which self-propelled colloid particles form regular crystalline arrays despite having only repulsive interparticle forces. So we talked about liquid crystals last year, I think, and how they can be applicable to biology. You have particles at different scales. You can model bacteria, collective behavior of bacteria that way, but also other types of particulates in actual crystals or in things that approximate crystals. And so this is the kind of system he's talking about. When the particles are dispersed and separate, each one gets propelled from place to place. So when you disperse them through some means, they get propelled all over the place. On the other hand, when the particles are jammed together in a dense array, the randomly oriented individual propulsion forces acting on them average out to zero, and the non-equilibrium driving produces very little motion. So when we talked about liquid crystal biology, one of the features of that is this idea of a jamming phase transition, which is where you go from having a bunch of particles moving around, interacting, to this jammed state where they're all sort of fixed in place because there are too many of them in a space. So if you think about, like, if you have a bunch of particles or in some sort of membrane, if it's enclosed in that membrane and it's very packed very tightly, there's a lot less opportunity to move around than if that membrane contains a sparse collection of those colloids, sort of those particles. They can move around a lot more there.
In cells, if the cell elongates, it becomes more likely to be a fluid cell body than if they're all regular and shorter or more regular.
Yeah.
If you elongate them, they tend to move more like a fluid. So,
yeah. Yeah, they actually change state. Yeah. But he's also saying that there's this turbulence that gets introduced as well, with if you have a driving energy that's not only greater but has a non-equilibrium fluctuation too.
Yeah, and you'll get vortices, which I think are really neat.
Yeah. So this example that we just talked about describes something he calls low rattling. Low rattling behavior is first identified through the observation that the same non-equilibrium system can be capable of exhibiting randomly diffuse exploration of configuration space, meaning that you have that things are exploring this configuration space, they're moving around, and they're taking different configurations. And so you can, just through diffusion, you can explore this space, and this is done randomly. This non-equilibrium system then can also explore the space just through this driving. So it's driving these particles around the space. They're moving at sort of a random rates, and they're moving randomly in random directions. But they're actively exploring this configuration space. So if there are things that are interesting in this configuration space, they tend to move towards those things. So if you think about like structure, if you have things that aggregate in certain locations, that can be done through random processes, just through some driving force forcing it into exploring its space more. And then you can also get orderly motion, or you can get stasis as well, depending on what part of configuration space it happens to be in, while exposed to the same external drive. So the external drive is driving this exploration, and it can drive the system into these ordered states, depending on what's going on. This effect has now been established in a variety of simulated and experimental systems, ranging from disordered spin glasses and mechanical networks to robot swarms. So the basic principle is a choice of external drive, so like this time-varying force, will determine the amplitude and degree of diffusive randomness in the local motion. So then he gives a loose analogy to machine learning, because of course he does. A neural network is a complicated function with many parameters that are mapped to output quantities. You're training the network, you're selecting the parameters so that the input-output mapping optimizes the configuration. And that's an analogy to what's going on here with this low rattling, self-organization by low rattling. So this is kind of what he talks about. And then he's talking about life far from equilibrium, which is where you're applying this to biology and statistical mechanics. So now you have living systems that can be organized using this low rattling principle, this low rattling measure, and you can characterize it. And talking about cells being far from equilibrium in at least 3 crucial respects. So they're far from chemical equilibrium, they're far from conformational equilibrium, they're conformational disequilibrium, and then they're in locational disequilibrium. Since macromolecules are arranged across space, but they're done so in ways that are tightly influenced by driven transport processes. So this means that this living systems are very amenable to this sort of approach and this sort of explanation. So this is an example of 2 assembly states here this is sort of what he's talking about. So, you have something in this state, say, for example, the external drive energy pushes it up into this state, and you have these multi-states that can, something can transition from one state to another. This is very simple, sort of complex systems, a complex systems diagram sort of explanation. Then he talks about spatial diffusion. So you have molecules that can diffuse over space, and this has a similar effect, drives it into new states, into different aggregations. You also have chemical reactions that, with the driving force of energy, can be driven to new states, and so that holds. And this is spatially dependent, so this is very amenable to the cell. And then he talks about some new opportunities for this, and they're kind of the same thing as what we talked about with things in the cell, these different types of subsystems in the cell, chemical systems, physical systems, that sort of thing. So that was that paper. I thought that was really interesting work. Now I'd like to go over some examples relevant to the Jeremy England paper. The first one being the role of actin filaments in wound healing, which shows self-organization. And the second being some drawings. We're going to talk about the fluxes of energy going into a system. So the first example is from this animation, and these are actin filaments that are forming a ring and then disappearing. So they're all over the place, and then they're pushed into this configuration of a ring, and then they disappear as the wound heals. So they need to be there temporarily, and they need to form a pattern in order for this process to unfold. So this animation is from the paper Coordinated Efforts of Different Actin Filament Populations Are Needed for Optimal Cell Wound Repair. So the abstract reads, cells are subjected to a barrage of daily insults that often lead to their cortex being ripped open and requiring immediate repair. So this is where a cell is ripped apart, the surface of the membrane is ripped apart, and the cells lose their integrity. An important component of the cell's repair response is the formation of an actin-myosin ring, which you saw in the animation, at the wound periphery to mediate its closure. Here we showed that inhibition of myosin, or the linear actin nucleation factors, diaphanous and or DAAM, results in a disrupted contractile apparatus and delayed wound closure. So these factors that are essential for this process, if you inhibit them, this disrupts this contractile formation. So you have this ring, and it closes down and then disappears. This is an important aspect of this process. We also showed the branched actin nucleators, WASP and SCAR, function non-redundantly as scaffolds to assemble and maintain this contractile actin-myosin cable. Removed branched actin leads to the formation of smaller circular actin-myosin structures at the cell cortex and slow wound closure. So you can have variations on this. The larger ring is more efficient than the smaller rings, but as we mentioned in the paper, we need to have this conformational, searching this conformational space and ending up with these patterns that actually do something. So we end up with either these large rings or these small rings in this case. Removing linear and branched actins simultaneously results in failed wound closure. So if we remove all of the actins simultaneously, we don't get wound closure. Surprisingly, removal of branched actin and myosin results in the formation of parallel linear F-actin filaments that undergo a chiral swirling movement. So this is where you have this ring, but it swirls around in a certain direction, orientation, to close the wound, thereby uncovering a new mechanism of cell wound closure. So again, even when we have this removal, we get this different type of wound closure that eventually forms. So these are all formations. These are all configurations of actin, these molecules, in different ways. And this is a product of searching that conformation or that possibility space, the number of possible states. Now I want to talk about a system, and we talked about a generic system where you have this energy flux. So we'll think about this as like a signal that goes up and down. And so this is the intensity of the signal of energy. And this is the external driving force to this internal system. We'll just have a bunch of molecules in here. We'll just put circles. It's a good example. So what we have here, and we've talked about this in the video, is these circles are at a certain density. And this energy, this driving force comes in and pushes these circles around in different directions. You know, if we're talking about Brownian motion, they're all kind of moving in different directions, and they're searching this conformational space for something stable, and that stability might be like a ring structure like we saw with the actin, or it could be like smaller rings. Well, these look like the cells, but the cells are kind of forming this packing, or you could even have a square or something like that. This is driven by the energy flux. So if we had a normal energy flux, something that wasn't really fluctuating over time, but it was basically uniform over time, we would get this sort of constant Brownian motion in this space, and these molecules would bounce around. And they may or may not find their conformational stable state. They might just bounce around infinitely. And that's, of course, what you see in a Maxwell's demon. You see in one chamber, you see molecules bouncing around, and they don't really form any sort of order. It's only when they go through the door to the other chamber and sort themselves that they form that order. And that's the idea of Maxwell's demon, and the idea there is that it defies thermodynamic law, that you can't simply have one chamber and another chamber and have a passageway through which these molecules sort themselves, unless there's some strong identity one way or another. And then we're beyond the realm of thermodynamics at that point. So this is a normal uniform signal, I guess is a better way to put it. Now let's think of a non-uniform signal, or a large set of fluctuations. So let's think of the energy signal as something like this. I mean, this is kind of a little bit different style than the sine wave I gave before, but this is highly irregular energy flux. So you have, large fluctuations at the top and at the bottom, and you have, states in the middle, and there's no character. It's non-uniform. So this sort of signal actually can do a lot to work to sort these molecules in this chamber. It could push them into rings. It could push them to the sides, to the walls. It could do a lot of things. It could disrupt when there's some sort of pattern that's already forming, so it could even disrupt that. The point is it would force these molecules into different states, or would force them to explore their space a little bit more. And so you have a situation where you have this space where the molecules are moving faster or slower than they normally would under standard Brownian motion, and you have this super regular movement. So that would be something that we would end up getting these kind of patterns where they end up engaging in these sort of weird behaviors. Molecules on opposite sides of the chamber, for example, might end up getting close to one another, things like that. So that's what we have for that example with the external driving energy. Now, I talked about jamming phase transitions before in the video, and let's take our chamber, and it's at a certain, it's at a certain sparsity here. It's not too sparse, but not too crowded. And the molecules can bounce around. It's like Brownian motion here. And that's all well and good. And we have the example we showed before. But this next example is where you get what they call critical jamming. And critical jamming is where you have molecules that are packed at a certain density so that they cannot move. And so something like this would be jammed. There's a critical parameter of crowding or of density that triggers this transition from these freely exploring molecules to this packed state where they don't really move around. And this is irregardless of the input energy, whether it's uniform or non-uniform. So a non-uniform energy might serve to break up this jamming if there is nothing else in the chamber. But if the entire chamber is jammed at a uniform density, then it can't really do much to push them around. It can just kind of introduce energy, and that may introduce other factors. So it may translate into forces between the cells or molecules, or it might translate into pushing them in different directions up and down. If you were in a three-dimensional box, if you only had a two-dimensional chamber that was square, then that would translate into some sort of energy to either the molecules or the cells or whatever these things are. So if you have local jamming, of course, it could break it up. But this is a very important point. So if you have this relatively sparse packing at the top, you're going to have these sorts of patterns that are able to be formed. If you have jamming, you won't necessarily be able to form these patterns, although you could, if you had local jamming instead of global jamming, you could force these apart and force these molecules into new configurations. So if you think about things that change their shape, or they move out of a stable state into another stable state, or move away from stability, there are a lot of different dynamical regimes that we might consider that would be explained by this external driving energy being a change agent. So if you have something that's, you said, non-uniform here, and it's coming in and affecting this packing, it's either breaking it up, or maybe it's enforcing jamming, because it's forcing all of these molecules to a certain part of the chamber. Say, for example, they have an electrical potential or a magnetic potential. And so, we can see that effect as well. So this is an interesting paper. I could give a number of other examples. This doesn't necessarily relate to the biology too much, except to say that if we think of these as cells or molecules like, actin filaments or something like that, then, it's a little bit easier to visualize. And then, taking this further out and thinking about networks. How are these things networked? And how does this network change with the introduction of energy in this way? So you can have these fluxes of energy that shift a network in different directions, whether they be maybe tensegrity networks, or networks that are organized by proximity, or networks that are organized by some other factor. Like a neural network where you have neurons that are next to each other, and you have energy that, maybe pulses of energy that modify their growth or modify their ability to send axons out to find neighbors, or even like activation energies, activation strengths. So I think that's all I want to talk about for that. Hope you learned something. Oh, hello, Sushmanth.
Hi.
I don't know if I guess it kind of recaps a lot of stuff that we know, but it also kind of explores potential mechanisms for explanation. Some of these things we've been talking about with physics and physiochemistry and all that. Oh, you're muted, Sushmanth. Did you want to say something?
Actually, I was seeing last year project DevoGraph actually.
Oh yeah.
I have some doubts during while reading it. I have gone through the work of G. Angley, I think so. In stage one, just small doubts. I just need a recap or confirmation from that. I'm just asking you. In stage one of DevoGraph, they are converting raw data of videos and images into some CSV file, right?
Yeah.
From those CSV file, we are transferring it into some graph embeddings. Is this process going on? I mean, that is the process, right? I mean, I'm a little confused while I was reading it. I had a meeting with Mainak and Mayuk also. They too did, like, they also helped me. They told, like, you can just go and ask Bradly. He will be good to help. Yesterday night.
Yeah, so the step one that we did last year was to use DevoLearn, the software, to generate like CSV files, which are basically the output of the. So you're analyzing data sets or images, and then they're analyzed, and there's an output of a CSV file, which gives you like the segmentation data from the images, the training data set that we have. Then that part is. Of course, we've been refactoring the software to make it optimal for these graph structures that we want to produce. So you get these segmentations of cells that are like cells with a centroid, and then those are positions that are given. Now you can build...
in stage...
Oh, go ahead.
It's in stage 2, actually, the parent cell and daughter cells are like, some cells are not connected. I mean, can I share my screen and show it to you?
Yeah, go ahead.
If you don't mind.
Okay, so this is stage 2. Yeah.
Yeah, stage. See, these are. I mean, can you see these cells are not project? They work like that.
Yeah. So
I mean, for every parent cell, daughter cell, right? And they have these pairings. For these cells, there is no pairing.
Could you scroll down a little bit? I can't remember exactly what this graph was. I think it might be labeled down at the bottom or up at the top. Yeah, I can't really see your screen.
Here he mentioned like the.
Yeah, he talks about this. I remember he generated these. Yes, I can see it.
Yes.
So yeah, I mean the thing here is that he's making different connections with this. So I'm not sure exactly what these connections represent. Usually, once we have the data from step 1, we'll have these points, and they can be related in space. Now they form a graph, but the graph doesn't have an absolute connectivity. So you can use different criterion to form the connections. So I don't know what criterion he was using here. I was trying to remember what that was, but basically the important thing here is that you have a bunch of points, and it's extracting potential networks. So these are just potential connections that they're generating.
Okay. Cool. I understood. I got some clear idea right now.
Yeah. Have you talked to Jia Hong about this, or just Mainak?
I talked with Mainak. Mike was also sitting beside him. He just explained him in the process. This is going on. And they were talking about like their GSOC work actually, what they had done in the DevoLearn. They explained me a lot yesterday night. I had a before meeting with them yesterday night actually.
Yeah.
After that, I have gone through the documentation of DevoGraph. It's pretty cool. I mean, it's interesting right now. I started learning this DevoGraphs, graph neural networks actually. I was watching some Stanford lectures about it. It's pretty cool. I think I will just see. I need to work on this. Like there are some connections, right? Some parent cells are not having some daughter cell connections. I think I was implemented correctly for different timestamps.
Yeah.
Is it?
Jiahong Lee is the person who you might want to message in Slack because he did a lot of the work on this step 2 last year. That was his project. It's part of the project. And so he really had worked through this. Yeah, what he did is he developed it from an existing paper and he refactored that code. So it's actually published in a paper too.
Okay. If you don't mind, can you share that paper? I think there is a I think there is a paper. Yeah, I've gone through it. PDF.
Yeah.
Cool. That's cool. I was thinking to start. Do you have any idea of how improving it or something like that? Same projects, because I think it is not completely done. Some parent daughter cells are not connected, right, to the parent. I mean, some graph embeddings are not completed.
Yeah.
Actually.
Yeah, I don't know. Like, I think it's going to be maybe incomplete to some extent because if it's taking it from a training set, it might not be able to recognize every cell. Yeah, so they have to do some work on that too.
Okay. Cool. That's it. Those roads are that for me. Anyways, thanks.
Yeah. All right. Well, thank you, Sushmanth. And let's stay on, have a dialogue about this. If you have questions, you can come to the meetings or talk about it in Slack. So we can help you out with this.
Okay. Yeah. Every Monday will be there, right?
Yeah, every Monday. Yeah.
Okay.
And there's a steep learning curve on this. So, so that's all for this week. Thank you for attending.